Before we get to the new content for today, I want to review a very key concept that I may not have emphasized in the previous sections. And that concept is structure and function. I think we talked about this during the identification of the carbohydrates lab, where the shape of a particular molecule will enable it to react with other molecules, or the shape of that molecule will provide a certain functional capacity for that molecule. So I want to go back and look at the carbohydrates and some of the lipids, and then tie that into what we're going to be looking at for today. So reviewing the carbohydrates, the first two carbohydrates that we looked at were monosaccharides and disaccharides. So for example, monosaccharides are sweet. Glucose is the example that's been given here. Disaccharides are also sweet, and sucrose is the example when you combine glucose and fructose together. You get sucrose, or table sugar. So both these molecules are very, very small compared to larger polymers, of course, and they are able to fit inside your taste buds. Therefore, you're able to taste them. The larger carbohydrates cannot fit into your taste buds, and so they have a different purpose. So the smaller ones have a purpose, and they are to indicate flavor. The larger ones will be in the next slide here. So we have monosaccharides, we have disaccharides, we have polysaccharides. The first polysaccharide we took a look at was cellulose. Take a look at the first slide here, or picture in the slide. You can see that the shape of the molecule is straight. And because it's straight, it's able to lie down flat, and therefore it's able to provide a structural purpose. Now imagine you have a single piece of paper. You can tear that single piece of paper very, very easily. But when you start to stack the pieces of paper, one on top of the other, imagine trying to rip now a phone book in half. It's going to be very difficult because each of those pieces of paper by themselves is weak, but together they provide a lot of strength and structure. That's how it works also with the cellulose cell walls of plants. They're able to lie down flat and form these strings and fibers, and therefore they provide strength to the cell. Take a look at the middle picture. The middle picture is going to be starch. It's lightly branched. Because it's branched, it's not able to lay down flat. Because it's not able to lay down flat, it has a different purpose. And it is going to be functional. It's going to be used for storage. It's the energy storage for the plant. Glycogen, which is the third picture, that is our storage molecule. We eat plants that have starch. We break them up enzymatically, digest them in our stomachs, chop them up into smaller glucoses, smaller hexagon rings, and then we reassemble them into highly branched molecules of glycogen inside of our body. Once again, these do not lay down flat, so they have no structural purpose, but they have a functional purpose, and it's to store the energy. Taking a look at lipids, we took a look at triglycerides. These are fats and oils that have long chains of hydrocarbons, the carbons and hydrogens. If you take a look at the first picture, this is a saturated triglyceride. It has all single bonds, and because of it, the chains are flat. And because of that, these molecules are able to lie down on top of one another, and it forms a solid fat. If you have a double bond, like you have in the second picture, it forms a kink in one or more of the chains. And that will prevent the chains from sitting down on top of each other. They cannot lay down flat. Because of that, they actually have enough space between them to have some motion, and they flow. So these are going to be unsaturated triglycerides, meaning that it's not completely filled with hydrogens, because if you look at the carbon-carbon bond, there are a couple of spaces, which I'm going to identify right now. There's a couple of spaces where hydrogens are absent, and that's because there is the double bond that's there. It provides that bend or kink, and therefore this is going to be an oil. So again, structure and function. So today what we're looking at are going to be steroids. If you look at this, the first steroid is going to be cholesterol. The second steroid is going to be the female hormone estrogen or estradiol. The third one is testosterone, and the fourth one is cortisol. We're going to focus on primarily the testosterone and the estrogen. Now, take a look at all four of these molecules. You should notice something a little bit familiar about them, and it's they have a very similar structure. They have 4 hydrocarbon rings. So they have 4 hydrocarbon rings on each of them. The examples are going to be testosterone, and if you look at the name testosterone, it even almost has the word steroid in it. We have estrogen, which is the female hormone, and vitamin D, and they're all derived from cholesterol. And look at cholesterol. Even cholesterol has the root steroid or ster in it as well. So cholesterol has sort of a bad name or a bad connotation to it. And the reason why is because we think of the bad cholesterol, but it's actually, we need to have some cholesterol in our bodies because it's converted into these molecules. If you're low on cholesterol, you won't be able to produce the testosterone needed if you're a male. If you're low on testosterone, you will not be able to produce the estrogen in high enough amounts if you're a female. Also, simply by going out into the sun, there's a photochemical reaction within your skin that converts cholesterol into vitamin D. And that's why people in regions such as ourselves and even further north, northern latitudes like Alaska, they have difficulty during the winter months and are vitamin D deficient. And that's because they're not getting enough sunlight, and that's not converting their cholesterol into vitamin D, which affects mood. So vitamin D is a good molecule, and it's derived from cholesterol. Now take a look at the difference between testosterone and the estrogen. Again, they have 4 hydrocarbon rings. Take a look at the ring, the D ring. It's exactly the same on both molecules. Take a look at the C ring. It's the exact same on both molecules. The B ring is very, very similar, except between the A and the B on the testosterone, there is a methyl group. And you'll notice that between the A and the B on the estrogen molecule, there is no methyl group. There's one other difference, and that's on the A ring. And on the estrogen, you have an OH, and on the testosterone, you have an O. What I want to show you is that both those molecules are very, very similar, except for slight modifications. And it's that one CH3 and the lack of an H. So what distinguishes the difference between a male and a female, the sex hormone, is 1 carbon and 2 hydrogens placed differently around this hydrocarbon ring. And that determines whether you're going to have male characteristics or female characteristics. Those are the steroids. At a little bit more at cholesterol. Cholesterol is not free floating inside of your blood. It's actually attached to a protein. And since cholesterol is a steroid, and since steroids are part of the lipids, when it attaches onto a protein, you have a lipid protein complex, or called a lipoprotein. So the cholesterol attaches to a protein in the blood, and it forms these structures called lipoproteins. And with these lipoproteins, there are 2 types. There's a good and there's a bad. Having a high cholesterol is not always a bad thing because it depends upon which type of cholesterol that you have. There's a lipoprotein called an LDL, a low-density lipoprotein. This is a bad cholesterol. This is the cholesterol that can get deposited in the walls of your arteries and can start to block them and clog them in a process called arteriosclerosis. So if you've ever heard of the narrowing of the arteries, what's happening is that your arteries are getting layered with these LDLs, and what it's doing is it's narrowing your arteries. What that's going to do is that's going to cause your heart to pump harder because it needs to pump the blood through a smaller channel. That could possibly lead to high blood pressure, also called hypertension. So that's the LDL. We don't want LDLs in our body because they have a negative effect on us. However, there is a good cholesterol, and it's an HDL, a high-density lipoprotein. This is the good cholesterol. The reason why it's good is because it goes in and removes the LDLs from your artery walls, and it brings them to the liver for destruction. So it'd be great just to go to the store and buy a bottle of HDLs and down that in one of your meals, and then you'll be healthy. But it just doesn't work that way. You have to naturally increase your HDLs. You can also do it with certain drugs that can increase the HDLs and also decrease the LDLs. But you can naturally increase your HDLs by eating a balanced diet and exercise. By doing those two things, your HDLs will automatically increase, and since they're increasing, it's going to automatically reduce and decrease the LDLs, which are the bad ones that are clogging your arteries. So by exercise and maintaining a healthy diet, that will keep your arteries clean. Okay, on to and switching to proteins. So we're leaving the lipids. We have the triglycerides as one type of lipid. We have the fats and the oils in that category. Then we also took a look at the steroids, and then particularly we focused on cholesterol. Now we're changing and looking at proteins. Now, protein composition has both carbon, hydrogen, oxygen, and nitrogen. When we took a look at the carbohydrates, they had carbon, hydrogen, and oxygen. When we look at the lipids, they have primarily carbon and hydrogen. So the new one in this is going to be the nitrogen. The nitrogen enables proteins to be built. Taking a look at this picture is going to show you a couple of amino acids. Take a look at the blue colored region, and that has a nitrogen and two hydrogens. It's very close to a molecule NH3, which is ammonia, and this side group of NH2 is called an amine group. If you look at the red shaded portion of the amino acid, the C with the double bond O and the OH beneath that, that is called the carboxyl or carboxylic acid group. So the name, the amino acid, comes from the structures that are attached to the amino acid. You have your amine group and you have your carboxylic acid group. Combine that together, you have an amino acid. Look at all 6 structures and you'll notice that they all have an NH2 and a COOH. They all have the blue portion and the red portion. They also have the hydrogen on the bottom of each of those molecules. What's different is what's called the R group, and that's the side chain. And it's the side chain that determines, again, how the amino acid is going to fold and how its shape is going to be represented. Take a look at the 4 amino acids. We have glycine, cysteine, pyrazine, and lysine. Each of them have a different side chain. One of them has an H, one has an SH and a CH2, and the other has a very larger complex, and the lysine has a hydrocarbon chain attached to an amine group. All those amino acids are different, and they're different because of side chains. This is the key, very similarly to where we saw the difference between the small carbohydrates and the larger carbohydrates, and we have flavor versus structure and function. If you're straight and you're a carbohydrate, you're going to be cellulose, you're going to have a function. You're going to have a structure. You'll be for structural purposes. If you're a lipid and you are straight, you're going to be a fat. If you're bent, you're going to be an oil. If you have 4 rings, you're going to be a steroid. So now we're getting to the main key concept for the proteins, and it's their shape. We have 20 amino acids, and each amino acid has a different side group or different R group. And it's the R group that's going to be determining the shape of the protein. Since the R group determines the shape, different amino acid sequence will create different combinations of R groups, will create different protein shapes, and will have different functions. Let me try to explain with the 2 different types of proteins that we have. So your amino acids can lay down in an orientation where they're elongated. And if they're elongated, they can form these string-like structures. Notice how you have in this muscle protein, you have 2 proteins. One of them is red and the other one is this purple structure. And it's an actin and myosin. These are filaments. These are fibrous and linear for structure. So just as cellulose, carbohydrate, polysaccharide is for structural purposes in plants, we also have these linear fibers called muscle fibers, and they're also for structural purposes. They provide and give us a shape and structure. The example for this would be our muscles. But there's another purpose for proteins, and it's a functional purpose. Just as we have the cellulose being a structural, and we have the starch and the glycogen being functional, proteins have structural and also functional purposes. The structural proteins are those that are linear, and the globular ones are functional. If you look at the root word of globular, you see the word globe. And let me represent this with a picture down below. I'm going to be drawing in a couple of things here. So this is going to be for functional purposes. So imagine that you have a hydrogen over here and an oxygen right next to it. These are two different amino acids. So I'm going to kind of follow along with, here's your amino acid chain, and these are formed up of anywhere between maybe 50 and a couple hundred amino acids to make this protein. And each protein will have a different shape. The reason why they have different shapes is because of their side groups. And the reason why they have different shapes because of their side groups, because their side groups form these interactions. The H will bond onto the O. Now imagine if you have a positively charged amino acid and a negatively charged amino acid. These are going to be attracted to each other. Amino acids with sulfur also called what are called disulfide bridges. And then you can have other interactions. We'll just say, again, this is maybe a hydrogen and an oxygen forming another hydrogen bond. And what it's doing is it's causing this linear protein to fold up in this particular shape. And it has this opening right here that a particular molecule can then fit inside, which is called the active site. So your proteins are either going to be structural or they'll be functional. In this case, this is a functional protein. So here's the structure. You have two amino acids. They're linked side by side, one amino acid or one peptide and a second peptide. They linked together to form what's called a peptide bond. And right here is the peptide bond in between the two amino acids. Here's the first one, here's the second one. And now we have two amino acids that are linked, so therefore we have a dipeptide. So in our next lecture, we're going to see more on the functional proteins rather than the structural ones. And the functional proteins are going to be our enzymes and how they work into breaking down molecules and building them up.